Next part. This is reverse transcriptase. So this thing up here is this thing over here. Great. Yeah, all right. Reverse transcriptase has got some parts. Yeah, so let's learn the parts. So look at this little animal up here, and they've colored in the parts very helpfully. So there's two big parts. The one part's called P66, the other's called P51. P66 is all the colorful stuff up here. P51 is the gray thing back here. P51's job is to hold P66 together and make it look good. So let's not worry about P51 so much. P66 has two main functions. So you've got the actual polymerase, and polymerases look like little hands. They have a fingers domain, they have a thumb domain, and they have a palm domain. And the fingers and thumb rather, will hold on to the RNA or DNA, whatever we're copying. Polymerases just look like this. They all look like this. And the palm is where the magic's going to happen. So that's where we're actually going to add nucleotides. The job of the fingers and the thumb is to grip the right thing and hold it against the palm. And I'm probably drawing on my hand as I am doing this. Yeah, that's it. And the way it actually works is like one hand clapping. What's the sound of one hand clapping? It's the same as the sound of HIV replicating. Yeah, I don't know, but it's a terrible sound. Ooh. The thumb up here and the fingers are actually going to open and close as the virus replicates. And so it's going to do this. It's going to like grip it in place and then open to let the RNA move on, and then grip it in place and then open to let it keep, feeding through, something like that. We do have a video of this. That was in the link at the top of the last presentation. We will watch that video in class. All right. So the job of the polymerase part is to take the HIV RNA and make a cDNA, complementary DNA copy. RNA, the copy of it is complementary RNA. RNA, the copy of it is complementary DNA. It's complementary because it has to be, because that's how it's made. Nevertheless, you will see this term a lot. cDNA. It's like a little c and then a big D and A. And that's all it means. Complementary in the sense that if there was a C nucleotide in the regular strand, there'll be a G in the complementary strand. It's a reverse complement. Okay, so we're going to turn RNA into DNA and then DNA into double-stranded DNA. But we have RNA and we copy it into delicious DNA. But then the thing is, now we've got DNA stuck to RNA, and what do you do with that? That's kind of nothing. It's like halfway between two things. So what the virus does is it's going to destroy the RNA so that it can make another DNA copy of the DNA. Yeah, right? RNase type H is this. That's what this RNAsh thing looks like. So RNase is an enzyme that breaks down RNA. It's like RNA A's, and they just squeezed the two A's into one and made it little. And then type H, there's a bunch of different RNases in the world. This was the, eighth one to be described, something like that. And so what RNase H does is, if it sees RNA attached to DNA as a, like reverse complement of each other, so a hybrid, RNA-DNA hybrid, it will destroy the RNA half and leave the DNA half alone. That's a very convenient enzymatic reaction, isn't it? Because this is how HIV clears off its template. It clears off that DNA after it's just made it by getting rid of the RNA that brought it there. Yeah. Not very loyal, HIV. Not very loyal at all to the RNA that started the infection. But there you go. That's how HIV rolls. Yeah. Can't tell HIV what to do. You don't know HIV. Yeah. Actually, you kind of do know HIV now. Yeah. And we're getting to know it even better, right? All right. So, HIV, reverse transcriptase has all these parts. And we talked about all the parts. The only part we didn't mention is the connection part. And so the connection part is like a web. So you get your RNase H over here, and you got your finger, thumb, palm polymerase over here. The connector part kind of flexes and bends between these two as they're working. And you would normally say, who cares? Why does that even get a name? Some of the most effective reverse transcriptase inhibitors that we have come up with bind to the connection domain. And all they do is lock into a little pocket in the connection domain and stop it from moving. Yeah, I know, right? Yeah, totally dumb. And these are things that they were just scanning random compounds, and they happened to find something that worked. And then after they figured out that it worked, they looked where it bound, and they thought, oh my gosh, really? There? That's not where it should bind. But yeah, it binds there, and it stops HIV by stopping it from being able to move. So it like locks it in place. So the HIV can't do what it needs to do. So that's good. All right, fine, done. Move on. Let's talk HIV, and we're going to talk HIV, and then we're going to talk HIV in class, and we are then going to end this little section. So HIV, here we go. This is the process, and I think this is going to be the only virus that we cover in this amount of detail, its replication. So every virus has some weird process by which it copies its RNA or DNA or whatever, both. And it does all sorts of weird things. There's a lot of weird proteins involved. There are so many steps. We're going to learn one. And that one is going to be HIV. Because we're trying to learn, everything about HIV. Yes, so you are HIV masters when we are done with this course. Or at least the first third of this course. So, here's how it starts. All right. HIV starts out bound to a cellular transfer RNA. So do you remember transfer RNAs? These are the things that you use in translation. So a transfer RNA is folded up like a little clover leaf, kind of like they show here. And at the end of it, it's got one amino acid. And so when you're actually making protein, the ribosome doesn't grab individual amino acids. It grabs a transfer RNA that has an amino acid on it. And it puts it in there, and then it pops the amino acid off, and then it throws away the transfer RNA like a discarded banana peel. Yeah. Anyway, HIV's there. HIV doesn't care if there is an amino acid on the transfer RNA. It just wants the transfer RNA for itself. And, yeah, if you were a transfer RNA, I can see how this might be attractive, right? So the transfer RNA is actually going to come in preloaded onto HIV. So remember when we were talking about Noah's Ark earlier? Yeah, you've got your 2 RNA copies. You got your 2 integrases on the RNA copies. You got your 2 reverse transcriptases next to the integrases on the RNA copies. And you've got 2 transfer RNAs. And it's a specific one for HIV-1. So let's call it like it is. Transfer RNA for the amino acid lysine number 3. And we'll make the LYS little. There we go. There we go. And there we go. Edited. Yeah. So for different retroviruses, they're going to do exactly the same process as this, but they're not going to use transfer RNA for lysine number 3, which is HIV type M, group M's deal. That's the deal. So, yeah. And really, does it matter? No. It matters because HIV has a little site in it, which is right here. It's this little red site, and it's called the primer binding site. And what it is a copy of the very end, the very tip of transfer RNA lysine number 3. And I'll show you how that gets there. But the first time it must have got there by accident. And then after that, it's there for a reason and by a definite mechanism. So HIV comes in with like a little Velcro pad. It's all of the nucleotides complementary to tRNA for lysine number 3. And now it's going to replicate. And so, yeah, I guess our joke of the day would be, why is a polymerase, a viral polymerase like Justin Bieber, The answer is that neither of them can really start anything on their own. They require a pretty good head start, like somebody already recording, the bulk of a track, and then they'll come in and they'll chuck in a verse or something on top of it. But yeah, they're not going to do a whole song. They're not anymore. No, not now. They're too big for this sort of thing. Yeah. And so that was pure slander about Mr. Bieber, and we apologize to him and his representatives, but it's absolutely true about polymerases. Polymerases can't start. Polymerases need a little head start, which is a thing called a primer. So a polymerase can extend a primer that's already there, but it cannot make a primer. To make a primer, you need a primase. Yeah. And so a lot of viruses, good working class blue collar viruses like a coronavirus, they're going to bring along their own primase. They're going to make their own primer from scratch. It's just how you do it down the docks with the other coronaviruses. HIV, though, lazy. Yeah, lazy virus. It's not going to make its own. It's going to steal, transfer RNA for lysine, number 3, and it's going to use that as its primer. How do you like me now, cell? Do you like HIV more or less after we talk about it? I feel like it's pretty much just the worst in every way. Just the worst. All right. So HIV just recruits one of these tRNAs from the cell. Just says, yeah, you're mine now. Yeah. And it's going to take it with it into the virion and leave the cell and bring it into the next cell and then use it as a primer. So all a primer has to be is a piece of RNA or DNA that a polymerase can then extend. And that is exactly what we do. So the next thing that happens is that we are going to extend that RNA. And reverse transcriptase is an RNA-dependent, reads RNA, DNA polymerase. So it writes DNA. And actually, it's also a DNA-dependent DNA polymerase because it can read DNA and write DNA too. It doesn't care what it reads, but it's going to write DNA, darn it, because that's all it knows how to do. So we are going to make the copy. And in this one, when you see a blue copy, slightly blue, like this one down here, that's going to be DNA. And when you see a green copy like this, that's going to be RNA. And when you see blue stuck to green, why, that's an RNA-DNA hybrid, right? Yeah, sure. Okay. Second step, HIV is going to cover its tracks. It's going to degrade the RNA that it just used to, that it just copied. And so the HIV RNA genome actually comes in with a cap and with a poly A tail so that you could translate it right away if you wanted to. But yeah, instead you're going to reverse transcribe it and it's going to do terrible things. So the cap and the poly A tail got there because of the way that this mRNA got made. And I just let the cat out of the bag. It gets made like an mRNA. So, yeah. Now might be a good time for our second Easter egg. Shall we? Shall we? Let's. All right. Second Easter egg is, tell me the PDB code. So the Protein Data Bank is the place where all the structures of proteins go to live. And so tell me the Protein Data Bank accession number. So it'll usually start with a number and then it'll have 3 either numbers or letters after it. So like 1BEN is probably something. I have no idea what it is. Yeah, but yeah, I'm sure it's something. And yeah, I want you to tell me the PDB code for the first reverse transcriptase structure with the whole enzyme in there. And if I'm thinking of the same one you're thinking of, it's from 1995, back in the day. Oh my gosh, this structure may be older than you are. It's probably older than you are. Too bad. Yeah. Too bad, you little kids. Yeah, younger than the HIV reverse transcriptase structure. Yeah. Oh my gosh. All right. Fine. Next step, because let's go through all the steps. And they're written here to help you study, but let's go through all the steps. Yeah. So we made that little blue copy, this little blue tag here. The gray thing that looks like a cloud, that's reverse transcriptase. And it's going to go out to the end where the cap is, and then it's going to stop because it has nothing left to copy. just like Justin Bieber. When there's nothing left to copy, he stops singing. Yeah, again, that's slander. Mr. Bieber's estate, please leave us alone. Yeah, we don't mean anything by it. It's illustrating a point. Then the next thing that happens is this whole thing that we just made. So RNase H comes in and it degrades all of the RNA that we were bound to. And now this is going to come loose. And we're not sure exactly how this gets transferred, but it may be transferred by reverse transcriptase. So reverse transcriptase is going to be still attached to the thing it just made, and it's going to go to the other end of the genome. And so the last thing it copied is a region called the repeat region, which is the little R that's right here. So there we go. So the newly synthesized DNA is going to be transferred. Oh my gosh, transferred with 2 R's. There you go. That never happened. And it's going to hybridize, which means bind to, like Watson-Crick style, you know, A to T, G to C, etc. It's going to bind to the other end of the genome. And it's going to bind to a region that is the same sequence as the repeat it just left. So the repeat is repeated twice in the genome, and it's at both ends of the genome. And this is why. So we're going to take this primer we just made down to the end. And so the HIV genome is going in this direction, just like the little arrow. So 5' to 3', that way. And so our new strand, we started by copying 5' to 3' that way. We ran out of room to copy, and so we picked it up, put it back here at the other repeat, and now, oh my goodness, we can now copy all the way to the end. Yeah, fantastic. All right, and so it's going to copy, it's going to bind to this repeat, and it's just going to make a complete copy. And that is called first strand synthesis. So we do the first part of first strand synthesis up here. We do the second part down here. So we transfer that first bit to the end and then run it right back to the beginning. And so now we have an RNA and DNA hybrid. So we've got an RNA and DNA, and it's got a little piece of DNA from the front of the virus that's now at the back, and it's attached to a big RNA from the rest of the virus. So neat and complicated, yeah, but not that bad. So the first 4 steps are first strand DNA synthesis, or if you want, first strand complementary DNA synthesis. Why not? And all right, second strand DNA synthesis is the next step. So once again, HIV, it's got to cover its tracks. It doesn't want you knowing it was once a lowly RNA virus. It's all shiny and DNA now. Yeah. So next thing it does is, of course, it's going to erase all that RNA. Just degrade the heck out of it. RNA gone. RNA wiped out. Except for a little piece. There's a little piece kind of in the middle, which is called the polypurine tract. Now, purines and pyrimidines, do you remember them? I have my way of remembering them. So the pyrimidines are like C and T. So I imagine a cookie tower with little Chips Ahoy's, I think, in my mind, stacked up to make a little pyramid. It's the cookie tower pyramid. C and T are pyrimidines, which means that A and G are purines. Yeah. So who cares about A and G? Well, this is a polypurine tract. So it's a whole bunch of A's and G's. Thing about A's and G's is nothing. They're just bigger. They're nucleotides. And so the A's and G's are going to bind to their complementary C's and T's. And you're going to get a stretch here that is going to be, for whatever reason, kind of invisible to RNase H. RNase H is really bad at dissolving polypurine attached to polypyrimidine. Yeah, what can I say? So that's its one weakness. That's its kryptonite, if you like. Yeah. So we're going to leave that little bit. And wouldn't you know it, that little bit's going to serve as our next primer. So now this piece of RNA that was left over, the polypurine tract, is the only thing that's left. And the reverse transcriptase is going to walk down here, get on the end of it, because just like that next Justin Bieber album, he's going to get on the end of whatever song is popular, and he's going to make it better, isn't he? He's going to make, well, maybe not better, he's going to make it longer. Polymerases make things longer. Again, please don't sue me. Yeah. So the polymerase is going to extend its little primer that's right here and run it all the way to the end until it runs out of stuff to copy. Yeah, great. And then, what do you know? It's going to come in handy again. And so our little tRNA binding site region up here, that is going to be the last thing that we copy, because after that there's just the tRNA. And so we're actually literally making a copy of the beginning of the tRNA here, and that's how the beginning of the tRNA complement gets into the genome in the first place. So we're going to peel this off once again. Reverse transcriptase is going to, RNase H is going to finally finish dissolving this thing, but only after it's used it as a primer. Yeah, so, yeah. Then we're going to pick this up, and we're going to pick this whole thing up, and we're going to bring it back up, because the last thing we copied was the complement of it was the primer binding site, where the primer binds. And so, yeah, we're going to bring that up here, and we're going to set it on there, and now the little red arrows are complementary to each other, and so the primer binding site serves as the primer. And so now 2 reverse transcriptases are going to take over and finish this job. So this one's going to see this as a primer, and this is a template, and it's going to finish it to the end. This one's going to see this as a template, the long strand, and this little thing as its primer, and darn it, it's going to finish it all the way to the end. And so 2 reverse transcriptases, or 1 reverse transcriptase, getting to the end, and then moving over and then finishing the other thing, we are going to get this job done. We're going to get this built, and so we're going to end up with a double-stranded DNA version of the virus, and that's called the provirus. Think like professional virus, yeah. And the provirus is going to be a little different from the actual virus. So the actual virus started with the repeat area up here and went all the way down to the repeat area, and that was it. The provirus has this extra thing called the unique 5' region, and it has a unique 3' region, which is past the repeat. And so it got those, and you can go back up here and see the steps where it picked those up. Yeah, it's got those, and so now the provirus has this whole cassette, which is, cassette just means region, with the U5 region, the repeat region, and the U3 region. So U5, repeat 3 at both ends, like a duplicate, an exact copy. And so that's how the provirus is different from the actual viral RNA that comes in. It's just got this piece, and so the U5 was right up in here, and it gets copied, and the U3 was right up here. Yeah, they both just get copied, and they both end up on both ends. So there you go. There you go. All right. Thus ends our reverse transcriptase saga, and reverse transcriptase is going to take us a while, so we'll take some time with this. Thanks very much.